Good morning, students. This is your biology teacher. In our earlier class, we have learnt about microorganisms under the title of friend and foe. Already we have learnt about the different kinds of microorganisms, whatever may be existed around us. While studying about the microorganism, we have observed in the water, in air, and in the soil. Along with these three, in other microorganisms and in plants, in animal also can be alive, these microorganisms. Among these microorganisms, some are useful microorganisms and some are harmful microorganisms. Already we have learnt about them. And where are the microorganisms present and where does the microorganisms can be alive? The microorganisms can be alive in everywhere. Already we have discussed about that. And before going to where does they live, we have to notice what are the different kinds of microorganisms. The microorganisms are majorly 5 kinds. One is bacteria, second one fungi, protozoa, virus and algae. So we can find all this everywhere, means in water, in soil, in air, and in animal body, in plant body, in hot spring, in deserts, cold condition and acidic conditions, and everywhere. Even in another microorganism bodies also can be alive, these microorganisms. So let us discuss about where does these microorganisms can be alive by the activity, or where do microorganisms live. To find out these microorganisms, we have to collect some soil into a beaker. Later we have to pour some amount of the water into them. After few minutes, that entire soil should be dissolved in the water and settled under the water in a beaker. And after that we have to collect one drop of water from the upper surface of water, and the collected drop of water put on the slide, glass slide. Later this glass slide keep under the electron microscope. Then when we have observed along with our eye, some microscopic body should be moved on the slide. What happened there? So the soil is also have different kinds of microorganisms, whatever may be there, that is maybe bacteria or virus or fungi or algae, whatever may be there, they should move on the slide. So that is one activity. And another along with that, to find out that activity to in water. So the water is also has different kinds of microorganisms. How can we identify the microorganisms in the water? To identify the microorganisms in the water, we have to take some stagnated water from the pond or from the drainage, and among them we have to take one drop of the water and put on the glass slide. Later this glass slide has to keep under the microscope, electron microscope. Then when we have observed that in that drop of water is also should be contained some moved bodies. While collecting, we can't identify any microorganisms in the drain water or in the stagnated pond water, but when we have keep under the electron microscope, we can notice several microorganisms, those are moving in the water. And along with that, another example from the air. The air is also contain microorganisms. The water has microorganisms, the soil has microorganisms, and as well as the air is also have microorganisms. How can we find out the microorganisms from the soil, from the air, as well as in the water, as well as in the air is also can be find out. To find out the microorganisms from the air, we have to keep some vegetables or pickle or bread at the outside of the room temperature. So immediately what happened, that material should be contact with moisture from the air. So along with moisture, the bacteria or virus or fungi or algae, whatever may be there in the air, they should try to damage the vegetables or pickle or bread or whatever we kept outside of the room temperature. After 2 to 3 days, they should make some patches and gradually the material should be spoiled. So we have to collect a few amount of the material from the spoiled and keep under the microscope. Then when we have observed under the microscope, they should show different kind of growth on the bodies of the vegetable, on the bodies of the maybe pickle and bread. And sometimes we can easily identify it without using any aid or microscope, the patches, however it may be there, as like a filament like structure, the growth of the filament should be appeared on the fungi, as like a fungi, mushroom, however is there. So they are belongs to fungi family. Here mainly what are there, the microorganisms should be present in everywhere is there. What is meant by everywhere? Through the first experiment we can find, we have found the microorganisms in the soil, and the second experiment we have found the microorganisms in the water, and the third experiment we have found the microorganisms in the vegetables, pickle and bread. So what are different kinds of microorganisms? The different kinds of microorganisms are bacteria, fungi, protozoa, algae and other different kinds of microorganisms. Let us discuss about that. Already we have learnt about that bacteria. So what are bacteria? Bacteria is a unicellular body. Already I have taught about the bacteria. What about bacteria? Is a unicellular prokaryotic body. Unicellular prokaryotic body. What am I said about the prokaryote? Which organism doesn't have the nucleus? They are called prokaryote. Pro means without, karyote means nucleus. Those cells, those organisms do not have the nucleus, they are called prokaryote. So bacteria is also belongs to prokaryotic microorganism. Here bacteria can be alive maybe sometimes alone or sometimes maybe colonies, by making colonies. These can alive in all kind of organisms. Even though in some bacteria, these bacteria can be alive in plants, can be alive in animal, can be alive even in human being is also can alive by obtaining food material, by making symbiosis method. Here everywhere is there. These are presented everywhere, bacteria. And also what is that, these bacteria, some are harmful bacteria is there and some are useful bacteria is there. We know very well about that useful bacteria and harmful bacteria. Example for that useful bacteria, we are using some bacteria in the kitchen to make the milk into curd, and as well as which is meant for lactobacillus bacteria, and another bacteria for the degrading of the garbage, and another bacteria in the cleaning of the society, and another bacteria is using in agriculture to get the nitrogen from the soil or from the environment, as like that. So some useful bacteria are there and some harmful bacteria is also there. So mainly what is there, when we have observed the internal part of the bacteria, the entire bacteria has some jelly like material is there, jelly. So it has the DNA will be there. There is no RNA in bacteria. The bacteria contain only DNA is there. What is meant by DNA? DNA means deoxyribonucleic acid. It has only one chromosome. Human being has 23 pairs of the chromosomes. We have 23 pairs of the chromosomes, but the bacteria has only single chromosome is there, only single chromosome. So these bacteria should be shows different shapes and different structures. What are the different shapes and different structures? So some are round in nature, some are cylindrical, some are spiral. What are being round in nature? They are called Quacky bacteria. Round bacteria belongs to Quacky bacteria, and cylindrical, a tube like structure or cylindrical bacteria belongs to Bacillus bacteria, and Bacillus and spiral bacteria, those have the spiral like structure. Binding spiral is there, as like that. Some bacteria is there, they are spirilla. So we know now different kinds of bacteria around us. So here how the microorganisms reproducing. Generally we know reproduction. What is the use of the reproduction? Reproduction is using for the perpetuation of their race on this planet. So here different kind of reproductions we know. In earlier class we have learned about the different kinds of reproduction. What are there? One is sexual reproduction and another one is asexual reproduction. So here most of the microorganisms should be follow the asexual reproduction by binary fission. What is meant by binary fission? Division of the microorganism. division of the microorganism along with nucleus or protoplasm. So in this binary fission, they do not need fusing of male and female gametes. Without fusing male and female gametes, these microorganisms can be multiply and increase their number. So mainly what are the bacteria and microorganisms has been increasing their number by the binary fission only. So here useful bacteria, let us discuss about useful bacteria. So in our daily life we are using some useful bacteria to convert the milk into curd, that is called lactobacillus bacteria. What is the shape of the lactobacillus bacteria? Bacillus bacteria. So previously we discussed about the bacillus bacteria. What is the shape of the bacillus bacteria? Cylindrical bacteria. So we are using cylindrical bacteria in the preparation of curd. Preparation of curd or conversion of the milk into curd. And another example for that useful bacteria, cleaning of environment. What is meant by cleaning of environment? In our daily life we have been using different kind of commodities, among them some overleft material, whatever may be thrown out from our house or room. So that should be decayed, maybe whatever we have to say, garbage or waste metal or heap, whatever it may be, that should be turned into soil by cleaning bacteria. What are cleaning bacteria here? Ideonella bacteria. The Ideonella bacteria can be degrade or decompose all the garbage, all the waste metal, whatever we have been overleft in our daily life. And as well as another bacteria, agricultural industry. So as well as cleaning of the society, as well as in our kitchen room, however helping some bacteria, as well as in the agricultural department is also some bacteria has been helping to get the required material such as nitrogen and etc. What is the bacteria using in agricultural industry? Rhizobium bacteria using in agricultural industry to trap the nitrogen and to supply the plant. And another bacteria, lactococcus bacteria, not a bacillus bacteria, lactococcus. So this lactococcus bacteria using in idli, rava, dosa, cheese etc. What about lactococcus bacteria using only to make the cheese from the milk. And for the fermentation of idli, rava, and whatever the dough we are preparing, different kinds of material, we are using lactobacillus bacteria. What is that? Lactobacillus bacteria. So let us discuss about preparation of the curd. So we know very well about that. In our daily life we are adding some amount of the butter or curd into the lukewarm milk. So it has to be lukewarm or warm, because to improve the population in the milk, it has to be need some temperature. So we have to provide some temperature to the milk. After adding some amount of the curd or buttermilk into the lukewarm milk, the lactobacillus should promote the formation of curd. How the lactobacillus can be promote the formation of the curd? So if we had a few amount of the lactobacillus bacteria along with curd, that number can be increased within few hours by the presence of lukewarm. So while then what happened, the lactobacillus should be released some amount of the enzymes into them. So through that enzymes, what happened, the milk should be converted into curd by multiplying the number. And as well as we are using these useful microorganisms by the preparation of the cheese and pickles and bacteria. So here curd, for the preparation of the curd we have used some lactobacillus bacteria, and for the preparation of cheese we have used lactococcus bacteria, and as well as for the preparation of wine or something we are using useful bacteria in our daily life. Later we will discuss about what are the useful bacteria present more than this, and what are the harmful bacteria present than this. Thank you.